Assalamu alaikum dear students. Today we will discuss advance in MLT MCQs part 1C. The first step of PCR is dash. Option A denaturation, B annealing, C primary extension, and option D none of these. So the correct option is A denaturation. The PCR technique was developed by option A Keremulis, B Kohler, C Milstein. So the correct option is A Keremulis. Subscribe now and press the bell icon. Never miss an update from MLT Technologist. PCR is used to dash. Option A amplify the gene of interest, B construct RAPD maps, option C detect the presence of transgenes in an organism, and option D all of these. So the correct option is D all of these. All of the following are thermostable polymerases except dash. Option A take polymerase, B Ment polymerase, C DNA polymerase 3, and option D polymerase. Correct option is C DNA polymerase 3. In apheresis, one of the separated portions is withdrawn and the remaining components of option A transfused, B retransfused, C discarded, option D wasted. So the correct option is B retransfused. Sanger DNA sequencing is also called as dash. Option A pyrosequencing, B chain termination, C Mcqs. Correct option is B chain termination. Sequencing DNA means determining the order of the 4 chemical building blocks called dash that make up the DNA molecule. Option A enzymes, B proteins, C bases. Correct option is C bases. With the addition of an enzyme DNA polymerase, the primer is extended until a dash is encountered. Option A DNTP, B enzyme, C soil, option D water. So correct option is B enzyme. DNA sequencing refers to the dash. Option A technique refers to determining the sugar sequence in a DNA molecule. This is Sanger sequence in DNA molecule. Option B technique refers to the determining the base sequence in a DNA molecule. All of these. So the correct option is C all of these. In Sanger sequencing, since the terminating nucleotide can be distinguished by color, all 4 reactions can be performed in a dash. Option A single tube, B double tube, C 3 tubes, and option D 4 tubes. So the correct option is A single tube. The enzyme used in the Mcqs-Gilbert method for P32 labeling of the DNA at the end of 3' end is option A polynucleotide kinase, B alkaline phosphatase, C exonuclease, and option D amylase. Correct option is A polynucleotide kinase. Which of the following is a chemical nucleotide sequencing method? Option A Sanger method, B Mcqs-Gilbert method, C. PCR, and option D. Automated sequencing method. Correct option is B. Mcqs-1C method. How many different types of chemical treatments are required in the Mcqs-1C method? Option A 0, B 2, option C 3, and option D 4. So the correct option is D 4. The disadvantage of post-Sanger sequencing method is option A, they are more costly, B, they require the construction of a clone library, option C, they are more time consuming, option D, only short nucleotide lengths can be read at one time. So the correct option is D. Only short nucleotide lengths can be read at one time. Guanine specific cleavage in the Mcqs-1C method is done by using option A, formic acid, B, hydrazine, C, dimethyl sulfate, and option D, piperidine. Correct option is C, dimethyl sulfate. The principle of Sanger's method relies on option A, use of chemical for base specific cleavage, B, use of DNTPs for chain termination, option C, use of DDNTPs for chain termination, option D, use of P32 for chain termination. So the correct option is C, use of DDNTPs for chain termination. The samples in Sanger method after reaction are separated using option A, AGE, B, PAG page, option C, PFGE, and option D, 2D gel electrophoresis, and option E, FAGE. So the correct option is B, PAGE page. Which of the following describes pyrosequencing, the post-Sanger sequencing approach developed by the 454 Life Sciences Corporation? Option A, a series of enzymatic reactions produced light with an intensity proportional to the number of nucleotides added to the growing DNA strand. Option B, a clone library of DNA fragments is constructed. Option C, DNA fragments are attached to a glass slide in a way that they form hairpins. So the correct option is A. A series of enzymatic reactions produces light with an intensity proportional to the number of nucleotides added to the growing DNA strand. So, DNA sequencing is done by dash. Option A, Mcqs-1C method, B, Sanger dy6kj method, option C, both A and B, and option D, Watson and Crick. So the correct option is C, both A and B. HPLC stand for option A, high pressure liquid chromatography, B, high performance liquid chromatography, option C, highly placed liquid chromatography. Correct option is B, high performance liquid chromatography. Thermus aquaticus is the source of dash. Option A, take polymerase, B, vent polymerase, C, both A and B, option D, primase. So the correct option is A, take polymerase. Which of the following is a chemical nucleotide sequencing method? Option A, Sanger method, B, Mcqs-1C method, C, PCR, and option D, automated sequencing method. So the correct option is B, Maxim-Gilbert method. Subscribe now and press the bell icon. Never miss an update from MLT Technologist. PCR is used in dash. Option A, site-specific recombination. B, site-directed mutagenesis. C, site-specific translocation. And option D, both A and B. So the correct option is B, site-directed mutagenesis. PCR is dash. Option A, DNA degradation technique. B, DNA amplification technique. C, DNA sequencing technique. Option D, all of these. So the correct option is D, all of these. Thank you for watching my videos.